So now that we have an understanding of some of the basics, let's review those things we were talking about at the beginning of the class. The Human Genome Project was completed in 2003. The goal was to sequence all of the human genome and completely map genes to chromosomes. So far, they have identified 20,000 to 25,000 genes in the human DNA. And this was an amazing discovery because before this project was completed, it was believed that a human would have about 100,000 genes. And it totally rewrote the books about one gene, one protein hypothesis. And now we know that it's one gene, many proteins, depending on how genes are spliced during their processing. So the human genome is made up of about 3 billion chemical base pairs. This information has been stored in databases and it's readily available to anyone in the world who wants to use it. And it has allowed for improvement of data analysis, and people can find some sequences and go back and compare. It's helped identify many, many Mendelian diseases, but now we're at the next step. What can we do with this information? So we've cloned the genes. Does that mean we could make a man? We have the sequence, we have the location of the sequence, but it's going to take a lot longer to understand what each gene codes for and what protein it makes. But it is still possible, because once you know the gene, you can know what the protein is made, and then with all this technology you can actually fold the protein into figure out what the structure of the protein is, and then try and figure out what that protein does. There's a lot of ethical, legal, and social issues, and how can we protect individuals from discrimination based on their genes. In 2008, they passed the Genetic Information Non-Discrimination Act, which should protect against some discrimination. However, if you listen to the news, just this week the Supreme Court voted to allow for DNA samples to be taken from any suspects that have been arrested in crimes, not found guilty, but suspects, and then they're able to compare their DNA to known DNA in banks to see if they were actually guilty of past crimes, especially heinous crimes like rape and other murders where they have some DNA evidence, but unsolved crimes. The Human Genome Project has a huge economic impact, and just showing that it costs about $3 billion to sequence the human genome, but they predict already 796 billion in economic impact, and created thousands and thousands of jobs in the world today. It really has been a genomic revolution. So what about medicine and the new genetics? Well, the first big thing is, if we know the sequence of a gene, a faulty gene that causes a disease, can we fix it? What about drug design? Can we design better drugs, more specifically based on a gene sequence and the protein structure, so that we could maybe have drugs with fewer side effects? Gene therapy was obviously one of the most exciting, but still so far not as successful as we would have hoped. The best example is the cystic fibrosis gene was cloned in collaboration with a lot of scientists around the world in 1989, and here we are more than 20 years later with no cure for the disease yet. We have been successful in some gene therapy, but very few and far between. The genes that we've been successful in are the genes that are present, expressed in bone marrow, because there we could actually take bone marrow from a patient. We could insert the gene directly into the place where it is expressed, and then we could actually get those bone marrow cells back reintroduced into the patient, and hopefully they would colonize. So here's some of the results. They've tried this on 6 severe immune deficiency patients. They've actually had success in 2 chronic granulomatosis disorder. These are disorders due to bone marrow misfunction. And they've had 1 hemophiliac that they tried to cure. Hemophilia is a genetic disease of factor VIII. It's not being produced properly, and that patient died of other complications. So the numbers are not that good as we would like to see. However, there have been some success stories. For example, the first gene to be cloned and expressed in very large quantities in bacteria was the insulin gene. And by 1978, we were expressing the human gene for insulin in bacteria and reproduced in large amounts for diabetics. So this is pure human insulin made from a human gene by a bacteria because the genetic code is compatible. Some of the success with vaccines, making edible vaccines, they've had some success in potatoes, but people don't really like to eat raw potatoes. They prefer bananas. So what they're doing is adding this virus, an altered form of the virus, into the banana sapling. It becomes part of the permanent plant and it expresses its virus proteins, which it then uses to those proteins would then create an immune response in anybody eating that banana. Up until now, our problem is getting this through the gastrodigestive system, because as we've spoken many times in class about the problem with our stomach is it's a great pit, acid pit, and so any protein that we're going to put in there is going to get broken down. The advantage of, and why we're pursuing this, is because the cost is a few pennies versus about $125 per dose. Genetically modified food is here, and it's probably here to stay. The first tomato was called the Flavor Saver tomato, and it was the first commercially grown, genetically engineered food to be granted a license for human consumption. One of the ways you can figure out if your food is genetically modified, when you look at the price lookup code, it's going to start with a 9. If it's organically grown, it'll start with an 8 if it's genetically engineered. Organically grown starts with a 9, and usually there's just 4 numbers. So a conventionally grown banana will be 4011, an organic banana would be 94011, and genetically modified would be 84011. There is no marking up on any of our products like corn that have been put into other products. For example, a cow that eats genetically modified corn the meat is not labeled that it ate genetically modified corn, and that's probably what people are up in arms about. Here's an example about adding vitamins into rice, so you could in transgenic rice. It has been approved in China since 2009, but they're still not mass producing it. If you think of countries like China, which has a food shortage, it becomes a huge advantage if you can modify the food and make it more viable for those people. And in England, they was experimenting with rice to produce normal human blood proteins such as factor VIII, which is used in diseases such as hemophilia. So if we can get something to grow and produce a gene, it's to our advantage. And of course, genetically modified corn has been used in the United States for at least a decade. It's in all of our food. It's fed to the animals that we eat. And there's starting to be a big movement in this country about genetically modified food. And you'll probably see the name Monsanto in the news a lot because we do not label genetically modified food in this country. Europe has been a lot more strict about genetically modified food than the United States. So what about designer babies? Would you want to alter the genes of your potential unborn child? We have the potential to do it, but how do we know what qualities will be useful in the future? What if we added a height gene, but in the process created an enzyme deficiency, so you'd end up with an unhealthy child anyway? So the biggest problem with recombinant DNA when we're trying to insert genes is getting them into the correct cell at the correct time and having all the correct switches to make that gene be expressed. The closest would probably be embryo selection and analysis of embryos prior to implantation, that those embryos are viable and that you've got the gene where you want it to be. But this is still cost prohibitive and something that we'll probably see more about in the future. Bacteria that clean up oil spills, so we're genetically modifying microbes to make them more efficient at metabolizing oil. And then, of course, some of the biggest news came out in 2010. Craig Fenter was one of the scientists who was part of the big Human Genome Project, but he was in the for-profit part of it. But he has created a self-replicating synthetic bacteria cell. What he did is he's modified the genes. He's assembled a genome of a successful bacteria, but he didn't create it from nothing. He put that chromosome into a basically ready machine. He put it into a bacteria that he had removed the chromosome from. So it's still a landmark achievement. It's going to be interesting to see if we can make useful microbes from scratch with none of any of the harmful things that some bacteria do, and maybe even to make products like vaccines and biofuels. So watch out, watch the space for what's going to be coming your way in the future. Of course, cloning, there's a ban on such experimentation. Is it safe? What it would actually be? The problem with cloning yourself is you're just going to have a genetically clone of yourself, but however old you are, that clone is going to take that many years to catch up to where you, and you could never recreate the same environment that that individual was in. Dolly the sheep was the first successful sheep cloned in 1997. She wasn't a success story. It wasn't easy. It took 276 attempts before she was born, and sadly Dolly died at a premature age of 6 years. The cattle breeding program is already huge with cloning, using frozen embryos of their prize meat-producing cows, and also the production of human hormones and proteins in transgenic cows so they can produce milk with an added protein have already been successful. None of these experiments are cheap to do. The glow-in-the-dark cats, this is all based on the technique of using a gene that came from a jellyfish. Then that jellyfish does bioluminescence, and now the scientists have cloned that gene and inserted it into unfertilized cat eggs, so that as a result, after fertilization, the cats may be resistant to feline immunodeficiency virus, but they also glow fluorescent green under special lights.